So what's the difference? Yes.
Active uses ATP and passive doesn't use ATP.
Good. Active uses ATP and passive does not. Does not require energy. What else?
So active is going from low to high concentration, and then passive is going from high to low.
Good. Low to high and it's going from high to low. So molecules move from high concentrated area to low concentrated area. In active transport, they move from low concentrated area to high concentrated area. So in order to do that, it requires ATP. What else does it involve? What? One more. Yuki.
Passive transport uses protein channels, and then active transport uses protein pumps.
Good. Okay. So these are transport proteins, but by function they're different. This is pump and this is channel. Pumps are, the word pump is like it's pumping something. It's grabbing something from one side and like actively putting it on the other side. So the pump is what uses ATP to transport material from low concentrated area to high concentrated area. Alright? Over here, the channel, protein channel, does not use ATP, but molecules just move down the concentration gradient. They just flow with the concentration gradient, high to low. Alright, I'm going to use a slideshow to go over some of the things that we have already talked about, and then go on from there. Alright, this is posted online, so you can follow along later. Alright, why don't you all just look at this screen and you can take notes later. Okay, unless you're taking notes on your computer, please have it closed. Okay. Alright. So here's the summary of passive transport. No energy is supplied or used, so no energy is required. Molecules move using their own kinetic energy. The kinetic energy is like what energy that molecules have. So they just use their own energy and they like bounce off, they collide with each other, and that's how they spread. When you spray the spray in one corner of the room, those spray particles spread throughout the room. Okay? The particles are bouncing off. They are colliding with one another, and eventually they keep colliding, and they spread through the rest of the room. Okay? It uses their own kinetic energy. Now, molecules go from area of high concentration to area of low concentration. So, in other words, we can say the same expression in this way. Molecules move down the concentration gradient. Moving down means it's like going from high to low. So moving down the concentration gradient is same as saying this. Alright? So moving high to low, same as saying moving down the concentration gradient. Okay, the gradient means difference, the concentration difference. So there is area of high concentration of whatever, an area of low concentration. Moving down means it's going from high concentrated area to low concentrated area. Right? So the word here, concentration gradient, means there's a difference in concentration. So there are two sides. There's high and there's low. It's going down, so going from high to low. Right? Okay. Now, when the spray particles have evenly spread out, those particles they continually move. They're continually colliding with one another. But because they're evenly spread out, there is no net difference in their rate of movement. In other words, how it smells in one corner of the room is same as how the other part of the room smells, because there is same amount of stuff like spread out in all parts of the room. Okay, so we say that the equilibrium has reached. It has equaled out. The concentration is equal or evened out in all areas. But here it says molecules stop diffusing. This just means that the concentration is equal. It's evened out. All right. But I actually like like this word better, dynamic equilibrium. Dynamic equilibrium. So it's equal, okay, evenly spread out, but it's dynamic equilibrium. In other words, the particles that have spread out, they are constantly moving. They're still moving, colliding with one another. They're still like active, but the rate of movement from one part to the other is no different. That's the same. So dynamic means they're still moving, but it's evened out. So like it's equal in concentration, right? But we always forget to think about that. Okay, when equilibrium is reached, do the particles then stay put, or are they stationary? Do they stop moving? No, they still move around, but the concentration is equal in all parts. Yes?
Like for example, if you're in a room that the molecules can't escape from and you spray a perfume or something, like how does the smell start to fade?
How does the smell start to fade?
Yeah. If the molecules are still in.
I'm not sure. I mean, I'm not sure. They sort of like, maybe they, the whatever is adding the smell only lasts that long. And maybe they evaporate, or I don't know. Maybe they're so spread out that we can't recognize anymore. Yeah.
Do they fall?
They could land and fall and whatever.
Yeah.
But just idea of the word diffusion means they're spreading out, right? They're diffusing, they're moving. Equilibrium means it's equal in concentration. But just don't forget, these particles are still moving. And so think in back of your mind, okay, it's really dynamic equilibrium. They are still moving around, but it's equal in concentration. Okay? All right, I'm gonna go over it because that slide was same as what we already mentioned. So here are types of passive transport: diffusion, facilitated diffusion, osmosis. So we'll go in that order. Diffusion means movement of molecules from high to low concentration. So it's also called simple diffusion. It just means it's going, the molecules are moving from high concentrated area to low concentrated area. And that's just like logic. That's like normal. That's what we experience all the time, right? When you spill water or when you like spray something, they just start to spread out, diffuse from high to low concentrated area. This is diffusion. That's also diffusion. This drop of dye will eventually like spread out and diffuse across this box. So is the red dot, okay? But this involves a cell membrane. Now, if this membrane is permeable to the red dot, then the red dots will diffuse out of the membrane. So whether these red dots will come out depends on the permeability of the membrane. Okay, so remember the cell membrane is called semi-permeable, selectively permeable membrane or semi-permeable membrane. So that word indicates, okay, it's semi-permeable. Some things are able to pass through, other things cannot. So it depends on the permeability of the membrane to the molecules, all right?
So it depends on
Permeability. Here's another example. If you put a drop of dye in water, they will eventually start diffusing. Like if you put milk or creamer in coffee, or it starts like spreading and they diffuse. All right. Again, the blue dots. Whether they will move across the membrane depends on the permeability of the membrane. But if the membrane is permeable to the blue dots, then which direction will the blue dots move?
In.
In. Good. All right, in. Now once equilibrium is reached, so after a period of time, you come back and look at it. Okay, so let's say equilibrium is reached. Okay, so the concentration is same on both sides. Now, okay, do you think it will have the same number of blue dots in the cell as outside the cell?
No.
No. Right? No. Right? Okay, why so? Yeah.
Wouldn't it just continue to move?
It will continue to move, but overall, right, the net amount in the cell versus outside, will the number be the same? No, not necessarily. Okay, but when we say the concentration is equal, what do we mean then? Yes.
It's generally spread out equally all over, like everywhere.
Okay.
Not just the cell, but also the other spaces.
Okay, so the concentration is same on both sides, but could we expect to have same number of dots? Not necessarily. Yeah. Sunika?
I was gonna say like the ratio is the same.
The ratio is same. The ratio. All right. Notice that the amount of water outside the cell is greater. Okay. So to make the same concentration or same strength, let's say like 10% salt versus and 10% salt. 10% salt solution here and 10% salt solution. Now the concentration is same. There's both 10%. But in terms of how much water and how much salt is in the cell versus how much water and how much salt is in here, it's not going to be the same, right? Obviously, you will have a lot more water and more salt to bring it to 10% salt solution here than here, right? So the ratio of volume of water versus salt is equal in both directions, but it's not necessarily the number of particles, okay? All right, which direction will these purple particles move? From A to B, right? From A to B. So please use words like, okay, side, one side, and to which side, for side A to side B, because it depends what we're looking at, okay? Now, that also depends on the permeability of the membrane. We assume that the membrane is permeable to purple dots. So in that case, particles will move from A to B, okay? All right, facilitated diffusion is diffusion using protein channel. Okay, so the only difference between diffusion and facilitated diffusion is that the molecules move across the membrane through protein channel. Okay. So in other words, with our earlier example, diffusion, okay, particles just move across the cell membrane. They just move across a phospholipid bilayer. They don't require a special transport protein to move across. Now, I am assuming that these hexagons are too big to pass through the spaces between phospholipids. Okay, they're too big. Or they may be also very largely polar items. Okay, the membrane is mostly non-polar because of the phospholipids. Okay, so non-polar things that are small and polar will have easier time crossing the membrane without the help of proteins. They will be able to move in just easily. But things like this, which is too big and they may be polar, so they can't just move across. They have to go through special protein, and that's protein channel. So protein channels don't use ATP to transport them. It just is like open channel. It allows them to diffuse, but it's specific. This protein is designed to transport this only. It can't transport like a circular molecule because their shapes don't match. This protein is very specific. Okay. Next is a picture of cell membrane. So these cubes will go from this side to that side. Okay, high concentrated area to low concentrated area. That's also facilitated diffusion. Okay, anytime you say protein channel and it's diffusion, okay, then this is facilitated diffusion. Osmosis is our third type of passive transport, and this is diffusion of water. Right? We want to pay attention to how water moves because cells exist in aqueous environment, aqueous like watery environment, and they respond to the type of solutions that they sit in. Okay? They constantly react and respond to their surrounding. Right? So what type of solution it is matters a lot to cell. So we want to look at, okay, how does the solution affect the cell? How does water move in or out of the cell? What will that do to the cell? All right. So the study of just the movement of water is called osmosis. Okay? Now water also moves from area of high concentration to low concentration. Okay? Where there's more water to where there's less water. Like if you have a beaker of water and the beaker then is an area of high concentration of water, and when you spill it to the table, okay, water spreads throughout the table, so it spreads from high to low, like high concentrated area to low concentrated area of water, right? Not of other things, of water. Okay, aquaporin is a protein that helps transport water molecules. So aquaporin is also an example of facilitated diffusion, or it's a protein channel. Okay, protein channel for water. Okay, so some cells have aquaporin. All right, just take a look at this slide. You don't need to memorize it. Just understand how these factors affect how the molecules move across the cell membrane. For example, concentration of molecules. So basically, one side might have more of that particular substance than the other side. So the concentration matters. If this side has more than this side, the direction of movement will be flowing this way. But if it's the opposite, the flow of molecules will go this way, right? So which side has more things dissolved in it? Size and weight. How big the molecules are. If they're super small, they may be able to just pass through the cell membrane easily. But if they're big, they will not be able to cross the cell membrane. Also, like the chemical nature, is it hydrophilic kind or hydrophobic kind? Is it nonpolar? Because if it's nonpolar and small, they will have easier time passing through the phospholipid. But if they're big and they're polar, then they won't be able to pass through. Okay? They will probably require transport protein. Okay? Charge of the molecule, that's positively charged or negatively charged charges on the molecule, or maybe they don't have charges. Okay, so those things affect the passage of molecules. Okay, now we're going to look at active transport. Yes, question?
Have you put this on the Google Classroom?
I think
Yes.
I did this morning, yeah.
Oh no, it's on part of the unit material.
Unit material. So unit material has the slideshows, and video and resources have videos and recordings of the lecture.
Thank you.
Yeah. Okay, let's take a look at this second question. What is so cool about the structure of the cell membrane? In your explanation, explain why fluid mosaic model best fits the cell membrane. So, can you discuss with your neighbor what is fluid mosaic model and why is this a good model that describes our current understanding of cell membrane?
Wait, I wrote
You know,
I don't know where this fluid.
What?
Wait, I wrote
Wait,
this
wait,
fluid
second
mosaic
question.
model.
How this
I wrote it somewhere.
Second question for
Oh.
model.
What is so
Because
cool
it's
about
like
the structure?
flexible and moves around.
We'll just
Then
discuss.
it has proteins inside it, and that is the best because.
wait, so we don't need to write it.
You don't need to write
Oh,
it.
here it
Okay,
is. It is fluid because it's smooth. Best fits. Fluid.
It's like a mosaic because of all the different parts, like the proteins and
Oh.
the phospholipid head, the phosphate heads of the phospholipids, and such.
Oh,
And also
since
the
the cell
glycogen.
membrane is composed of many parts that are constantly in motion.
Right. So
Yeah,
the
that's
motion part is the fluid, and many parts is the mosaic. Are we good? All right. Before we talk about active transport, we have few more things to talk about for passive transport. Okay. So we've mentioned that the type of solution that the cell is sitting in is very important because they respond to their surrounding. Okay. So we'll talk about solution. Here's a key point why we are studying the type of solution is here. Cells exist in an aqueous or watery solution, and the function of the cell membrane is to regulate the movement of substances into and out of the cell in order to maintain homeostasis. So what cell membrane does help with maintaining homeostasis. And that is about how it's adjusting or responding to the type of solution that it is sitting in. Okay, so the external extracellular fluid, the environment is water, watery environment. Also, what's inside the cell, that's also fluid space, filled space, right? So there are solution in the cell and solution outside the cell. So we need to pay attention to that. Okay, just terms. Okay, let's get these definitions straight. Solution is a mixture of two or more substances. Now, the Kool-Aid, okay, the powder being dissolved in the water, that makes the juice. Okay. Now, what is being dissolved? What is being dissolved is solute. Okay. What is being dissolved is solute. And what is doing the dissolving, such as water, is solvent. Okay. Now, the ratio of solute and solvent determines the concentration of the solution, right? How much sugar versus how much water, right? Those two things, the ratio of that determines the concentration. Okay? So, for example, like 100 ml of 10% salt solution versus 1 liter of 10% salt solution. Well, concentration is same, right? But in order to make 1 liter of salt solution, 1 liter of 10%, you have to take 1 liter of water and take more salt to bring it to 10%, okay? Over here, we only need 100 ml of that. Okay, so you take 100 ml of water and put in, smaller amounts of salt to bring it to 10%. Okay, but concentration is same. If you were to drink this or taste it, like the saltiness is same. Okay, but the volume is different. But the ratio of water to salt is equal in both. The ratio of salt and water is equal. That's why it's 10%. Okay. So what is doing the dissolving is solvent. What is being dissolved is solute. Now, how do you guys remember those two words? Which one is for which? So some things like this, it's gonna be confusing, right? Later you're gonna, okay, which one was which? I just can't remember. I gotta come up with something to help me remember.
I was gonna say solution because it's the end process, but that's one of the definitions of it.
So I don't know if this will work for you, but what I use is, okay, so there's solute and solvent, right? Solute and solvent. I know that solvent is like water, right? Like something like water that's doing the dissolving. And in our alphabetical, the order of the alphabet, B and W are closer together. Sorry, it might be more confusing, but this is how I remember. Okay,
My brain
U
just
and
broke.
V. Okay, B and W, they're closer together than U and W. U and W is further apart than V and W.
What?
Right?
Oh, I get
Oh,
it.
V. I thought
V
you
and W.
said D.
I associate
B
V
and
and W
The
together.
car.
That's what I'm doing. In my brain,
I thought
I
you said
associate V and W together. Because V and W are closer than U and W.
BMW. Sounds like BMW.
Yeah. Okay. Whatever.
That's how I would remember it.
Yeah. Because some things you just have to like come up with a way to remember. Okay, and keep it straight. Because this is a matter of like remembering something, right? You understand it, but you have to sort of remember. Okay, which one was solvent? Which one solute? Okay, V and W. All right. Water and solvent. Okay. There are other solvents. Okay, other solvents besides water, but water is a good solvent. So I remember. Okay,
I actually have
something
one,
like
but
water.
it's really bad.
Okay. All right. You don't have to use that. It's just something.
Like water solvent on your
Okay.
face. No,
All right.
no, no, it's terrible, but it.
If the
I don't know if it
membrane
helps,
is
like,
permeable to only water, right? This is water. If the membrane is only permeable to water, what will happen?
Oh my goodness.
Here's then my next question before you answer that. If the membrane is permeable to both the green dot and water, what will happen? Okay. Alright. Okay. Usually the the membrane is permeable to water anyway, all right? But permeability to the green dots, it depends, all right? So if the information says, okay, it's permeable to both substances, okay, then what? If it's only permeable to one thing, then what? All right, so look for those information. Okay, so what will happen if it's permeable to both things? Yes.
Like both will move
Both-
across,
both-
and both will eventually get into equilibrium.
Okay, but tell me specifically about water and the green dots. Yes.
Green dots will move from side B to A, and while water moves from side A to B.
Good. Okay, water moves from side A to B, and green dots move from B to A. Okay, over time, the concentration will be equal. Yes.
Like, for this salt, it's like on the diagram, it looks like salt is bigger than water. Does that mean it's gonna have a hard time getting through the water?
Right. So that's why I said, okay, if it's permeable to both things, okay, so then what? Okay, if it's not permeable, then the green dots won't move. They will just stay put. They won't be able to pass across. The only thing then will move is water. What will then happen is water, the movement of water will bring it to equilibrium. Okay, so we'll have more water moving from side A to B until it's equal in concentration. All right, what about here? What will happen over time?
What?
Wait, so
The water molecule.
do any
water
so only water could go
and then it
through,
will go
right?
high to low, wouldn't it?
Good. So it looks like the gaps between the gaps are too small for these colored circles to pass. So only water will go, right? So you will have net difference of water moving from this side to this side overall, right? In other words, what I'm saying net difference is that water will pass across in both directions, but you will have more water. The tendency is water moving from this side to that side. The rate of water moving from here to here is greater than the water moving from this side to this side. Okay, now let's talk about the types of solution. There are three types of solution: hyper, hypo, and iso. Okay, iso means same. So like isosceles triangle means it's got a triangle with two same sides. Iso means same. So the concentration of solution in the cell is same as a concentration of solution outside the cell. Okay? In that case, the movement of water is equal. Okay? They're moving at the same rate. Alright? So notice that the percent of salt solution is same 10% and 10%. Okay? So it's equal concentration. Now in hypertonic solution, here you can say that this cell, so ignore the arrow, this cell is sitting in hypertonic solution. The cell is sitting in hypertonic solution. That indicates the concentration of solution is greater in the surrounding solution than the solution in the cell. Okay, because cell is sitting in hypertonic solution. Okay, it's comparing something, right? It's comparing the concentration of here versus here. And it's sitting in hypertonic solution, so the solution in the beaker is greater than the concentration in here. So the cell is sitting in hyper. Hyper means over, more, like super, right? Hypo means the opposite, under, less, low, okay? So like hyperactive means like very active, right? If you have hyperthyroidism, okay, it's condition of the thyroid, hyper, means that's opposite of hypothyroidism. Their symptoms are total opposite. Hyper has to do with having too much thyroid hormone versus hypo, like not enough. Okay, so symptoms are like the opposite.
고조생기능.
Yes.
So the types of solutions around the cell, that only refers to the
Solution or surrounding solution?
the surrounding.
Good point. Okay, good point. What I might confuse you by saying this, but what's another way to say exact same thing? So one way is cell is sitting in hypertonic solution. What is another way to say the same thing? Oh. That sentence is saying in respect to what this solution is like, right? But if you were to say something in respect to this solution,
The solution in the cell is hypotonic?
yeah, the solution in the cell is hypotonic solution. So what does it indicate? It indicates the solution here is lower, the concentration is lower here than the outside. Right? But both sentences mean the same thing, right? They
Okay.
won't mean the same thing. So it depends how you're putting it into sentence. But to not confuse you, I'm using the same format. The cell is sitting in hypertonic solution. But that's the same as saying the solution in the cell is hypotonic solution. Hypo means under, right? So you sort of have to think about, okay, what is this scenario, okay? So hypo is the opposite. Okay, here, the solution in the cell is 20% salt, this is 10%, okay? That's hypo. Hypo means less. All right, let's take a look at an example and what will happen to each of the cell if they're dropped into different types of solution. This has lots of green dots, no green dots, a few green dots, all right? Now those are like animal cells. If you were to place them, how will each cell respond to the different types of solution? Okay, now here's my scenario. The green dots are not permeable to the membrane is not permeable to the green dots. So green dots can't go anywhere. So we're looking at movement of water, so osmosis, right? So what will happen to each cell? Okay. With your neighbor, why don't you discuss?
Wait, so
first?
the membrane is. Wait, so the outside is.
So
So it's
water
a
can
cell,
go
right?
through. Water can go through, but the green stuff can't. So
Water
the-
can go through?
yeah, water can go through. So then the
So-
first
So
one,
And
water
it'll be
would
like equal inside and.
what?
It
Not
would
in the
just go
textbook.
in.
The first one? What?
What?
I don't know. Wait,
No,
which
the-
one are you talking about right
The
now?
cell, like the water would just go in the cell.
The first one? Okay, if we're assuming the cell is a lower
Okay,
concentration
so
of green stuff,
I'll
then
let you discuss or think about it.
the
first.
water the water's gonna go out in the
Let's
first one.
First
right?
identify which of the solution is iso, hypo, and hyper. That depends. It depends on that. Which of the three is more like isotonic?
The last one.
The third one.
Middle or third?
Third. Because the middle one has 0, so
Middle
It-
one's
it-
hypo.
Yeah.
iso is the same.
Same, right?
Like isosceles
Strength of the
triangle.
concentration is same. So which one? The middle or the third?
Third.
Third.
Because the middle one has absolutely nothing.
Third.
Okay, so good. Now this is more like an iso, right?
Yeah.
To the cell. Because the cell contains solution, but it's not pure water. So there are things dissolved in the cell. Right? There are ions, small things dissolved in the cell. So what's in the cell is solution that's not just pure water. It's not 0%. Okay? So whatever is dissolved in the cell, okay, makes a solution not just water. So this is close to more, being an isotonic solution to the cell. Right? Since there's nothing dissolved in this water, this is hypotonic solution to the cell. This would make it hypertonic solution to the cell. All right? Okay, so now, then what will happen to each cell? Yes?
On the first one, since there's a really high concentration of green dots, the water in the cell is gonna go out in order to make the outside lower concentration.
Yeah, make it diluted. In order to make
Yeah.
this more diluted, you have to, water from the cell will come out. Okay, what will that to the cell?
Shrink.
Shrink. Cell will shrink, losing water. Okay? Over here, what will happen to the cell? first of water. Water moves in what direction? Should we depend on?
Water's gonna go in to dilute the insides of the cell to match the outside.
Good. Water will move into the cell to match the concentration, okay? And cell will gain water. What will happen to the cell?
Expands.
Expand, okay? It will expand because it's gaining water. All right. Here's a problem with animal cell. Once it gains too much water, that the pressure of the water coming in is greater than the pressure that the membrane can handle, the membrane will expand. At a certain point, it will rupture and lyse, okay, burst open. All right, that is called cytolysis, cell lysing. Cyto means cell, lysis means like lysing, break open or being split or cut. Okay, cytolysis. The cell will undergo cytolysis in hypotonic solution. Right? This is for animal cell, right? Because the animal cell doesn't have the cell wall protecting the membrane, and so the membrane is the only thing that could handle the pressure, but it can only handle so much. So when too much water comes in, eventually it will break. Right? Cytolysis, cell bursting, cell lysing. Right? Over here, when cell shrinks, losing water, that's called plasmolysis. So here's the word lysis, but it's really just shrinking, cell shrinking. Okay. Now over here, no change to the cell size. Water is moving in both directions at the same rate, doesn't change the cell size. The concentration is already equal. Okay. The following slides have some definitions, so I'm just moving. Yeah.
Wait, so it goes cells can go cytolysis in hyper or hypo?
Cells undergo cytolysis you have to think through that, because me telling you the answer will only make you memorize, but you have to put the pieces together. When cell is
because
undergoing,
less, and then when you fill it up,
right.
So
When
it's
cell is
hypo.
cytolysing or undergoing cytolysis, that means cell is gaining water, right? So water is coming into the cell. So only way for the water to come into the cell is if it's sitting in hypotonic solution. Okay, so in isotonic solution, it's already in equilibrium. There's no net movement of water. There's equal amount of water entering and leaving the cell. But in hypo, like your question, water moves into the cell, making the cell will swell up, and it could lyse or burst open. So this is true if the concentration difference is too great, like the difference is too much. Okay? If it's slightly hypotonic, then probably it's okay. The cell won't die. Right? In hypertonic solution, water moves out, cell shrivels and shrinks. So that's plasmolysis. All right, before I talk about that, so in plant cell, there's a little bit of difference in how the effect of these solutions. Okay, for a plant cell, they have their membrane is protected by the cell wall. Okay, so the cell membrane expands, okay, or shrinks, but it won't rupture because the wall protects it, and it will shrink, but it can easily recover. Right? It will shrink, lose water, and like shrivel up, but because of the cell wall, it can regain its shape if you place it back into normal condition. All right. So this the pressure that is exerted on the membrane because of water coming in and coming out, going out, is called turgor pressure. Turgor pressure is the pressure on the cell wall or cell membrane. Okay. Now, because of this, plants do best in hypotonic solution. They do best in hypo, not in iso. Right? Animal cells do best in isotonic solution because you don't want animal cells to gain water and like expand and burst, right? Whereas plant cells, they need water constantly. right? And so, in the, when you look at plants or vegetables, they look fresh, and that means these cells have maximum amount of water that the cells can store, okay? But if they're starting to wilt, maybe they are in more isotonic solution, but they don't look that healthy. In normal condition, this would be like onion cells, but when you add salt water to it, the membrane will shrink, it will lose water, right? So the outline of the cell wall remains the same, but the membrane starts to shrink, and you could even see that outline very clearly under microscope, right? So when you make pickles, right? You put cucumber, you add salt to the cucumber, and over time you see what happens.
Smaller.
It gets it sort of gets shrivel up and shrink, and what else you see? Water, right? Water around the cucumber. You didn't put water in there. Water came from the cucumber, okay, because of salt. Salt draws water out. Okay. One more. So here's a cell, plant cell, in hypertonic solution. If this was an animal cell, again, what happens to the cell? If this is an animal cell, what will happen to the cell?
Oh, it's gonna shrink.
Isn't Luna right?
Okay,
Wait, what
It's
is
gonna
Yeah.
shrink.
it will.
Yeah, she's right.
Okay, are you confident?
Yeah.
No.
It doesn't.
Because I thought hypotonic was expanding and
Well, think about this. All right, this
It-
is
above-
these are
it does
cells
shrink.
I guess.
Yeah, I think it does.
It does shrink.
Yeah.
It does shrink, okay? So if you're still a little confused, okay, take a look. I said the cell is in hypertonic solution. Which solution has greater concentration?
The water.
This or this?
Oh.
That. This, right? This. Okay, so you can come up with some just random number, all right? So if this is stronger in concentration, you can make it up. This is 70% salt solution, whereas this is 30% salt solution. Okay, now I see the number, right? That means 70% salt solution means, all right, it's got more. Let's just come up with, let's say 70% salt solution. This is only 10% salt solution. Okay, this means the content in this beaker is 70% of salt and 30% water.
Whoa.
Okay, this means 10% salt and 90% water. Okay, water diffuses from
High tone.
high to low. High to low. Right? High to low. Water diffuses from high concentrated area to low concentrated area. If water leaves a cell shrinks. Okay?
Yeah.
So you have to think which side has more water than which side. Okay? Since this is hypertonic solution, okay, the ratio of water is lower in here versus in the cell. Okay? So I hope at home, as you review, think through this multiple times, and after a while, it will be sort of second nature. Okay? So cell loses water. Okay? If this was animal cell loses water, and cell will shrivel up, undergoing plasmolysis, and it may just eventually kill the cell. The cell will not recover. But in plant cell, because because of the cell wall, water will come out. But as the cells shrink, only to a certain point. Because of the turbo pressure, keeps the cell membrane from only getting so small. Okay? So this is also called plasmolysis, but this cell could regain its shape. If you save this cell and put it in normal water or normal condition, it will regain. That's same for your plants. If you forget to water your plants and you feel like think, oh, my plant is already dead, and you, okay, like my last chance, I'll just try giving water one more time. Next day, it's like it revived. Okay, so because of the cell wall, these cells can recover, right? But if it were for animal cell, no. Okay, if it shrivels up to the point where it's like it lost too much water, you can't revive it. Okay?
Wait. So is that how humans die of dehydration?
Yeah.
So you can't, when someone's dehydrated, you can't hydrate themselves?
No, you can hydrate, but once your body has lost too much water, it's critical, right? Yeah.
And then when you lose so much, like a lot of water,
That puts a lot of the organs in like risk.
and then you die.
Probably.
And then you can't, wait, if it's at risk, you can't recover?
No, I mean human health, it depends. There's some medical events to help you recover. But if you were to take just one single cell and compare, okay, the plant cell versus animal cell, that's the difference. Okay.
What?
Now,
I just drank.
in hypotonic solution, what will happen? Water in hypotonic solution, what will happen?
Comes in and expands.
Water will come in and will make the cell to expand. Okay, again, it can only expand so much because of the cell wall, right? And so that will increase the pressure and it will actually keep the plant cell healthy. Okay? So plant cells do best in hypotonic solution. And that's what happens when you give water to plants. You don't like give caffeine or any like that, like ice, you don't give like juice or anything. All right? They give water. Plants do best in hypotonic solution. If this were to be in isotonic solution, well, that means the rate of water moving in and out is equal, right? But it doesn't keep the cell healthy. It sort of will look a little bit not so fresh, okay? Not so turgid, okay? So the word here is turgid. The plant cell is turgid in hypotonic solution, but in isotonic solution, the plant cell becomes flaccid, a little flat. Okay, so that's the difference between plant and animal cell. All right, in your textbook, it had the same diagram and it talked about plant cell and animal cell. All right, some of these words were not mentioned like explicitly, but it talked about them. Okay, so you can add these two words in your vocab list, even though they're not in the textbook. So turgid and flaccid. Let's do this one more time, okay? Iso in hypo, in hypertonic solution, okay? For plant cell, what will happen to cell? Okay, why don't you make a chart in your notebook and first write what will happen to plant cell, animal cell in isotonic solution, in hypotonic solution. Right now, please the movement of water for each movement of water, and the effect on cell size, maybe. Effect on cell size.
Wait, isotonic, hypotonic, hypertonic.
Solution. So the cell is in isotonic solution here, in a hypotonic solution, in
Or do you need
hyper.
dry it?
Yeah, make a chart in your notebook. Make a chart. So here's plant cell, here's for animal cell, and write about each one underneath the picture. Okay. You good? Do it together? Alright, let's do this together. So, how will water move here? In plant cell in isotonic solution. Where I would draw an arrow. There's no net movement. Okay, it's equal in the rate of movement of water. Okay. Equal. Okay. It's already in equilibrium. Okay, these are the words I think you should be able to use in your description. What will happen to the cell size? Well, for plant cell, it's going to appear flaccid. Okay, it's going to appear flaccid, a little flat. Okay. However, animal cell, so it's also equal. That's saying equal, it's already in equilibrium. But cell is good, happy. Okay? Animal cells, they do best. They do best in this kind of situation, best in isotonic. Okay? The cell size is same. No change. Right? It's fine. It's no change in cell size. It's not getting bigger, it's not getting smaller, it's same.
Wait, this is good.
Because the rate of movement of water is same, right, in both directions.
This is good.
Yes.
So what happens if humans drink too much water?
That's fine, you get rid of it, your kidney will filter them.
Oh.
Wait,
We're not single-celled organism.
Oh yeah.
wait, but isn't there such thing as drinking too much water?
Yeah.
Yeah.
So I don't understand. when
You
people
can
call
rarely,
me
you
single
can
cell.
rarely
That's
bring your
unicellular.
to drink it to that.
No, that's not even a roast because it doesn't make any sense.
That was the video where there's
If you
someone
think
drinking
about it.
water and their brain got too big.
What did
Yeah,
you
but
say?
you're ignoring all the signs that your body's
It's
telling you
unicellular.
when you're
Does that
drinking
make
too much
sense?
water. You're intentionally drinking.
Sure.
That just shuts down your
It
organs.
doesn't.
There's no such thing as like,
Stupid.
if you're drinking one more cup of water, it's not gonna be like
Yuki's.
your cells are not gonna be in hypo. I mean,
Don't worry.
because
it's
your
in the
body
basement.
is more
makes
complex
sense.
than simple cell. If you
Water-
want to take one cell and put it in hypo or in, then yeah, that is just one celled organism. Yeah, and you could kill yourself by doing any stupid thing.
True.
Yeah,
Yeah.
water intoxication is an actual problem, but that's to an extreme level.
Really weird though.
Yeah, it's rare. Like,
it's to an
your
extreme.
body will start giving you signs to stop that, and
Has anyone done it before?
Like puking.
Then what?
yeah, you can't
Water
really die. I mean
intoxication.
that you can't really like drink to the point where it will kill you.
Yeah.
You will stop drinking before you get to that point. Okay, occasionally, rarely, okay, someone could ignore all those signs and keep drinking to the point where your organs start shutting down. But that's like rare.
Yeah, bye.
Rare. Okay, plant cell in hypo, what will happen? Movement of water.
Wait,
Hypo?
no, into the
In.
cell.
Into.
Yes, hypotonic, so in. Okay. So I'm gonna draw a smaller arrow going out and bigger arrow coming in. Okay, that shows. Okay. All right. So water moving in, and plant cell will become turgid. Okay, it's like turgid, like pom pom. Pom pom. It's pom pom.
Yeah, healthy.
But right.
Because it can't. Because when it's in the.
I mean, it's not wrong. That's not wrong.
break.
Right.
not wrong.
Exactly.
So that's why it stays healthy.
Yeah.
Joseph went after
It
yourself.
stays. But for animal cell,
It will break.
it will break.
Is that a
Okay, so I'm gonna
Yes.
just repeat that there. Okay, so same, right? But cell, so the effect is cell will undergo cytolysis, possibly, right? It will start gaining water, increase in size, cell
Maybe for
increases.
animal cell?
Mhm.
Yeah.
Well, so this is going to the extreme, right? Cell will increase in size as it gains water. But if the solution is too hypotonic solution, and cause the cell to gain so much water, then it could burst,
What happens to the cell after it bursts?
dies. And the membrane is ruptured, so it's dead.
Wait, but like say like a cell is in your body that's dead, like where does that go? Does it just stay in your body?
Our body cells aren't gonna become like our red blood cells, maybe. Okay, our red blood cells in the blood because they are just in the blood.
I have dead cells.
So it could endanger our red blood cells, but like if you sit in, if you're bathing in like onsen water, like basically you're sitting in some hypotonic, maybe hyper, depending on what type of onsen water it is, right? Like there may be, okay, but our body, like our skin and everything, we don't just start blowing up, right? Our skin cells could
Wrinkle.
wrinkle up, but if you were to take like earthworms or like leeches or some other things, or even like single-celled organisms and put it in like salt water, it kills them. It's one way to kill bacteria or preserve food, right? Because if you make pickles, basically you're making the surrounding very inhabitable for like bacteria to grow, because their cell will rupture and die. They won't be able to grow and replicate again.
Is that why snails die when you put salt in?
Yeah.
Oh,
And
same thing.
leeches.
Yeah. All right. What will happen in hypertonic solution? Water in hypertonic solution, water will come out of the cell, right? So more water coming out than what's going in. Okay, so it will start shrinking. So water leaving the cell. Cell shrink. That's called plasmolysis. Okay.
Shrinking is plasmolysis.
And then, so,
Cell shrinking is called plasmolysis.
Mrs.
Yes.
What would you call it? Like, what would you call water from like a public pool?
Chlorine.
Chlorinated?
Chlorine
what?
in water.
What?
Well, I don't know because it depends what the cell is, what the solution in the cell is like.
So like if we were to go and then like we stay in there for a while and then when you look at your hands it's all like all wrinkly.
It's just like bathing, right? Your cells,
That's normal.
your skin cell will sort of like get water, like soaked in water.
Is that
And
happening?
maybe your skin cells are responding, reacting, but that doesn't mean that extra water is seeping in through our skin. Or we have this protected layer called skin that by sitting in water, we don't start blowing up. It's just dead skin cells that get water, right? The dead skin cell, the outer layer, those cells sort of get like they gain water and they start like sort of getting bigger, and so it makes our skin to like wrinkle up, but that doesn't mean, okay, the water has gone into my body, right? It's only in the outer surface. Okay, so this is same here, cell leaving the cell, water leaving the cell, and cell will shrink. Smaller arrow. Cell shrinks, and the effect here is that cell also, plasmolysis is undergoing. Plasma, that's same. Okay, but if we're to compare the difference, if you put this back in normal water, it's easier to recover. Okay, you can save this, but for animal cell, once it loses so much water, it won't regain its shape. Okay. All right.